In the previous lectures, I discussed the various processes involved in conscious behavioral actions such as language and reading. It's important to consider that in addition to conscious behaviors such as these, our bodies and our brains also engage in unconscious processes such as developing emotional responses to perceived stimuli. As we will see, the neurobiology of the emotional response system has many characteristics in common with the conscious response systems, such as language and reading. Recall from our last lecture that we used the example of how various parts of the brain are involved when we develop and perceive an emotional response to a stimulus. Various parts of the cerebral cortex, as well as parts of the subcortical areas, such as the hippocampus and thalamus, communicate with an area of the brain called the amygdala, illustrated here. The function of the amygdala is to receive sensory stimuli from other parts of the brain and decode them in the form of emotions. If we feel a certain type of emotion, whether anger, sadness, or pleasure, that perception probably came through the amygdala. As described here, the amygdala also plays a central role in our ability to sense potential danger in the surrounding environment and to mount a response to it. Sometimes that danger is in the form of potential physical harm. Sometimes it is in the form of something that creates a sense of emotional stress without the actual fear of immediate physical harm. In either case, our body has developed a mechanism to respond to the perceived danger or stressor using a combination of conscious and unconscious processes. The body has developed a very effective unconscious response to stress that involves both neurological and hormonal components. Once the amygdala has conveyed to the hypothalamus the presence of a perceived threat or other type of stressor, the hypothalamus triggers a series of steps in response to the perceived stress. The first of these is to communicate with the pituitary gland using specific hormonal substances referred to as releasing factors. When the pituitary gland senses these factors, it sends a message to the adrenal gland, the body's principal stress response mechanism. The adrenal gland, which is located close to the kidney, rapidly secretes into the bloodstream a series of hormones, principal of which are epinephrine, commonly known as adrenaline, norepinephrine, and cortisol, commonly known as cortisone. The level of these circulating hormones in the bloodstream is referred to collectively as the allostatic load. While a short-term stress response of this type is a protective response to perceived stress, chronic exposure to stress can lead to a chronically elevated allostatic load. Chronic elevation of one's allostatic load can be harmful in a number of ways due to harmful effects of prolonged exposure to stress response hormones. Chronic elevation of the allostatic hormones can have harmful effects on the lining of important blood vessels. The first step in this process is an inflammation of the cells that make up the lining of the vessel. As with any chronic inflammation, over time, these cells will start to lay down scar tissue in the walls of the blood vessel. This scarring leads to stiffness in the wall of the blood vessel, with corresponding increase in blood pressure and reduced circulation to the organs fed by the blood vessel. As a consequence of these changes, the risk for a range of chronic diseases increases, especially cardiovascular disease and kidney disease. Writing in 1999, McEwen and Seaman described the potential health consequences of chronically elevated allostatic load. They reported, allostatic load appears to be a useful construct for conceptualizing how wear and tear and increased morbidity and mortality are caused over long-term intervals, not only by the more dramatic stressful life events, but also by the many events of daily life that elevate activities of physiologic systems. They go on to say that allostatic load reflects the impact not only of life experiences, but also of genetic load. Individual habits reflecting such items as diet, exercise, and substance abuse, and developmental experiences that set lifelong patterns of behavior and physiological reactivity. These authors bring up the important point that chronic elevation in the allostatic load can be the result not only of inherited genetic traits or of stressful experiences or unhealthy behaviors as adults. They also suggest that early developmental experiences in infancy and childhood can have potentially lifelong consequences in this area. In 2012, the American Academy of Pediatrics released a report that summarized recent research on the ways early child adversity can affect both child health and subsequent adult health. Echoing the comments of McEwen and Seaman, the committee that conducted the study reported that longitudinal studies that document the long-term consequences of childhood adversity indicate that alteration in a child's ecology can have measurable effects on his or her developmental trajectory with lifelong consequences for educational achievement, economic productivity, health status, and longevity. Jack Shonkoff, the principal author of the Pediatrics Technical Report, made similar comments in an article published in 2009 in the Journal of the American Medical Association. Shonkoff reported that children from families and communities with low income and low educational levels may be especially vulnerable to the biological embedding of disease risk because of their disproportionate exposure to highly stressful influences such as neighborhood violence, dysfunctional schools, personal maltreatment, household chaos, and absent parents. The American Academy of Pediatrics report suggests that particularly toxic forms of stress experienced over prolonged periods can actually alter the anatomic structure and physiologic processes of a young child. The report states that toxic stress can result from strong, frequent, or prolonged activation of the body's stress response systems in the absence of the buffering protection of a supportive adult relationship. Such disruption may result in anatomic changes and/or physiologic dysregulations that are the precursors of later impairments in learning and behavior, as well as the roots of chronic stress-related physical and mental illness. How are we to understand the comment that toxic levels of stress experienced by infants and young children, quote, may result in anatomic changes and/or physiologic dysregulations that are the precursors of later impairments in learning and behavior, as well as the roots of chronic stress-related physical and mental illness, as those authors report. The human response to stress involves both conscious and unconscious perceptions of stimuli from the environment. The conscious perception of stress is mediated by the visual and auditory cortex as well as the frontal lobe, with signals conveyed to the basal ganglia and the adjacent thalamus and hypothalamus. A more rapid unconscious response connects the amygdala, hypothalamus, and pituitary gland. As with the more conscious activities, such as language and reading, these parts of the brain's unconscious emotional response system are connected together by white matter axons. As with other types of axonal connections, the more frequently these axonal connections are triggered, the more likely they will become myelinated and the more rapidly they will conduct their impulses. This brings up an important question. As a consequence of a more frequently activated stress response system, will a child raised in a stressful environment also develop an exaggerated response to stress with chronically elevated allostatic load? The pediatric report goes on to describe the specific ways that early and prolonged exposure to toxic stress can actually alter the structure and function of a child's brain. Exposure to stressful experiences has been shown to alter the size and neuronal architecture of the amygdala, hippocampus, and prefrontal cortex, as well as lead to functional differences in learning, memory, and aspects of executive functioning. Thus, the developing architecture of the brain can be impaired in numerous ways that create a weak foundation for later learning, behavior, and health. Hence, altered brain architecture in response to toxic stress in early childhood could explain, at least in part, the strong association between early adverse experiences and subsequent problems in the development of linguistic, cognitive, and social-emotional skills, all of which are inextricably intertwined in the wiring of the developing brain. This concept that the anatomical structure of neurons and their axonal connections can be affected by factors in the external environment is referred to as epigenetic imprinting. The DNA of the genes a child is born with will not change. However, the manner in which those genes are expressed in terms of neural structure and function can change as a result of these exposures. In terms of the stress response mechanism, a child born into and raised in a highly stressful family and social environment may develop an exaggerated stress response with profound potential effects on behavior both in childhood and later in adolescence and adulthood. This brings up two important questions that we discussed at the end of the previous lecture. First, if by adolescence a child has developed certain key neural pathways in a dysfunctional manner, is it too late to alter them to a more functional state? And also, can that child still learn new things and new ways of behaving once he or she becomes an adult? As with the previous lecture, the answers here suggest that the process of brain plasticity, that is, the subsequent altering of the neural structures developed during childhood, that plasticity is ongoing, giving each person the potential to change both neural structures and patterns of behavior internalized as System 1 thinking. That potential may slow down as adults age, but it does not go away. This suggests that even with those adolescents and young adults with some of the most self-destructive behavior, the potential for a positive intervention with long-term benefits is still there.